So Professor Rickinson, Professor Alex Wai, Professor Rick Wong, Professor Yi Ke Guo, who is not yet here, Professor Jiming Liu, Professor Kevin Leung, colleagues, alumni, and students. Okay, good afternoon. I am Catherine Hall, Assistant Professor from the Department of Chemistry. I'm the MC as well as the facilitator of the Honorary Doctorate Distinguished Lecture. Today, we are very pleased and honored to have Professor Alan Rickinson to give us a lecture on the story of viruses and cancer. To begin with, may I invite Professor Alex Wai, the President and Vice Chancellor, to deliver a welcome speech, which is pre-recorded. So, Professor Wai.
Professor Rickinson, distinguished guests, fellow colleagues, alumni, and students, good afternoon. I'm honored to welcome a world-renowned viral biologist and a leading researcher on Epstein-Barr virus, Professor Alan Rickinson, to Hong Kong Baptist University for this very special online distinguished lecture. HKBU conferred an honorary doctoral degree to Professor Rickinson for his outstanding achievements and remarkable contributions to society in November 2021. It's a tradition of HKBU to invite each honorary doctorate to deliver a distinguished lecture. Although the pandemic keeps us apart physically, we are connected here today through the online lecture. It's a real privilege to have Professor Rickinson here with us today. Professor Alan Rickinson received his BA, MA, and PhD degrees from the University of Cambridge. He's a leading researcher on Epstein-Barr virus, EBV for short, glandular fever, and several forms of cancer in humans. He's currently Emeritus Professor of Cancer Studies at the University of Birmingham, United Kingdom. Professor Rickinson once worked under the mentorship of Professor Tony Epstein, the discoverer of EBV, at the University of Bristol, United Kingdom. His own research encompasses a variety of aspects of EBV biology, including the whole cellular immune responses to virus infection, the virus mechanism of persistence within the lymphoid system of the immunocompetent host, and the nature of the virus association with several different types of human cancer. Professor Rickinson had made tremendous contributions to science in terms of elucidating the biology and immunologies of EBV infections, as well as the development of vaccines that fight EBV-positive malignancy. He directed the Institute for Cancer Studies at the University of Birmingham from 1983 to 2001, during which time the Institute developed it from a small non-clinical research department into a large research institute studying cancer genetics, viral oncology, and tumor immunology. Professor Rickinson's research team won the prestigious Translational Cancer Research Prize in 2015 for the development of a therapeutic vaccine that targets EBV-associated cancers. He was awarded the Marjorie Stevenson Prize by the Microbiology Society in the United Kingdom in 2008, and he's a Fellow of Royal Society and the Academy of Medical Sciences, and an Honorary Fellow of the Royal College of Physicians in London. He currently serves as the Chairman of the Disciplinary Committee Review of the Research Grants Council under the University Grants Committee. Today, Professor Rickinson is going to tell us a story about viruses and cancer. The EBV is widely distributed in all human populations and is carried by the great majority of people as a lifelong asymptomatic infection. Yet, this same virus is capable of driving infected cells into permanent growth and is involved in the development of an increasingly wide range of human tumors. Among EBV-associated cancers, nasopharyngeal carcinoma is one of the most popular tumor types in Hong Kong. This lecture will show you how the various factors can contribute to this unique cancer type. Professor Rickinson, please enlighten us. We look forward to benefiting from your wisdom and teachings, and I'm sure all of us will be energized by your talk. Thank you.
Thank you, Professor Wai. Now, please join me to warmly welcome Professor Rickinson. And the title of his lecture is Dangerous Liaison, the Story of Viruses and Cancer. So, Professor Rickinson, you may start whenever you are ready.
Thank you. First of all, my thanks to Professor Wei for his very generous introduction, and also to the university for giving me the great honor of an honorary degree, and also this opportunity to speak to you about a field in which I've been immersed for about 40 years. In particular, I'd like to thank everyone who is zooming in. If not, they're in the room personally. And I hope that you enjoyed the talk. Thank you for zooming in. Well, let's begin at the beginning and think about viruses and their relationship to cancer. I'm going to talk in very broad terms about the field which I entered, but which has been really going for about 60 years now, the link between viruses and human cancer. The next slide is not changing for me right now, but let me try again. Here we are. So, in a way, viruses and cancer seem like a meeting of polar opposites, because if you think about it very generally, viruses are self-replicating machines that use cells as factories for their own self-replication, so often killing those cells in the process. So think of a virus as something which enters a cell and uses it for its own purpose, like a parasite would. And here comes the yellow virus entering the cell. It takes over the machinery of the cell and begins to replicate itself and spread to a second cell. And the process goes on. It will spread to a third. But note that the first cell that was infected will die. And so the virus is spreading through living cells, but leaving a trail of destruction behind it. And you can see each step in the process. Eventually, this will be contained by the host immune response, but not without severe damage and often being done. And we're critically aware of that with the recent coronavirus epidemic, which would spread in exactly this kind of way. So viruses, self-replicating machines. Now let's think about cancer. This is, in fact, quite a different situation, where the cells themselves become self-replicating machines. And that goes through a process of sequential genetic accidents, which release them from growth control. So again, let's think about one such cell during the process of reproducing itself might pick up a bit of genetic damage, some mistake in the process of DNA replication between each cycle of growth. And that acquisition of that rare event might give that cell a competitive advantage against its neighbors. And so it begins to spread and grow. And one of those cells within that spreading clone might pick up a second piece of damage, which would again now look two red dots in that cell, give it a competitive advantage. And so finally, one of those in the competitively advantaged clone will pick up a third change, genetic change, and that might confer the cancer phenotype, the ability to grow uncontrollably. Now, in different cancers, the actual number of genetic mutations that have to happen by accident to produce cancer may be different, but in this example, I'm showing you just 3. So viruses and cancer do seem like opposites. In one, the virus is winning. In the other one, the cell is actually replicating itself. But when I began to think about a title for the lecture, I thought this doesn't really fit my view of what's going on. I much prefer the example of Dangerous Liaisons, a book which I've recently seen actually, in which the virus can be seen as an invader, but lives happily within the body, but only with a risk that the liaison might turn out to be really dangerous. Here's the book in its original form by Pierre-Ambroise Chaudelot-Decolle, and it's, of course, Liaison Dangereuse in French. It was written just before the French Revolution in 1789. And this book tells the story of the really disgraceful amorous adventures of the aristocracy at the time. No wonder there was a revolution. And they were misbehaving in serious ways. And this became a bit of a scandalous novel for revealing the kinds of attachments they would get up to. If you've not read the book, you should see the film. And here we have 200 years later, after Delacroix's book came out, Hollywood caught up with the idea and made Dangerous Liaisons. And you can look at John Malkovich here in the center. He looks like he might be a kind of epitome of a virus about to invade two unsuspecting young cells there, Glenn Close and Michelle Pfeiffer, and various amorous interludes happen during the film. But let me tell you, like a lot of illicit love affairs, it does not end well. So let's go on with our Dangerous Liaisons and think about how such a link between viruses and cancer might emerge. Well, cancers take years to develop, step by step. So if a virus was involved, what kind of virus would it be? Well, it would be a virus that naturally persists in the body for years because we need its presence there over several years, if not a lifetime, to be able to mediate its effects. And either it's a virus that acts indirectly to promote cell growth. So this is the first example that I want to show you of how a viral infection might, in the long term, give us a risk of cancer. And what happens when viruses infect a tissue that actually responds to damage by continual regrowth? So here's an example of what could happen. The virus goes in, begins to replicate, as we've shown, kills one cell, but see the adjacent cell to that death, now marked dark green, receives a signal saying, there's damage happening in this tissue, and it's my job to repair it. So I must begin to duplicate myself, to grow myself as a cell, and double up and triple up, and in that way restore the damage that the virus is doing. So it's a continual regenerative process in which the cells that are regenerating and trying to limit the damage are themselves growing and dividing. What kind of tissue could this happen in? Well, let me take you back 2,500 years for the moment to Hesiod, who was the first Greek writer to actually write down the oral tales of Greek myths that were well known in ancient Greece. And this one such myth is of the human Prometheus who stole fire from the gods and gave it to man. Zeus, the king of the gods, was absolutely furious at this theft and committed Prometheus to a terrible fate. He was chained to a rock, sliced open, and in this painting you can see him being chained, and an eagle, which is just appearing in the top left, is about to fly down and peck away at his liver and eat half of his liver one day, and then fly away. And overnight, the liver would degenerate and the eagle would come back. A particularly gruesome punishment and a pretty awful tale, actually, of which only the Greeks were possible of conceiving. So we know now from time immemorial that the liver is one such tissue. And sure enough, so what would happen when viruses infect the liver, what we see is that the regenerating cells that are trying to limit the damage done by the virus, they are at greater risk of picking up genetic accidents. So they themselves are not infected, but they're trying to repair the damage and build up their numbers. And as cell growth goes on, so these genetic accidents may happen over long periods of time and produce a malignant liver cancer cell. So immediately you begin to think, well, okay, let's think about the possibility of hepatitis viruses and liver cancer. Now let me introduce you to Baruch Blumberg, who was a geneticist and public health physician who was particularly interested in this question. He wasn't a virologist at all. He wasn't even a cancer biologist. He was interested in why are particular ethnic groups at high risk of particular diseases. That was his question. And at that time, it was very difficult to find genetic markers that distinguished one ethnic group from another. But he had a theory that if he looked in the blood at the proteins of the blood, he might pick up unique proteins which were unique in one ethnic group and not another. And that might give him a clue about the differences between them. And could the differences he might see in the blood, could they be linked to susceptibility to one disease or another? Well, it was a slightly crazy idea at the time, but he went ahead and took serum samples from various ethnic groups around the world. He had a huge bank of serum samples and began to look for protein differences within them. And in one group, he found a protein difference that under the electron microscope, under the very powerful electron microscope, he could see these rods and filaments in the preparation. And this, because this picture was seen very often in the blood plasma of Australian Aborigines, it just got the name Australia antigen. And what was this thing? Was it a protein or was it something else? Was it a virus? No, it wasn't a virus per se. It was some kind of protein. But actually, it turned out after a lot of work from Bloomberg and other groups that it was actually a protein that was overproduced by a virus that was in the liver and was pumping out a large amount of this surface protein called the surface antigen now. And this was a very important discovery when it realized that this was not a marker of genetic difference. It was the product of a virus infection. And so Australia antigen was discovered as the major protein of the hepatitis B virus. A very important observation because it allowed us to think about this as a marker for infection, and antibodies to that protein would be a marker of having got hepatitis B virus. And it was also a route towards vaccine development. So all of these were very important follow-ons from this chance observation. So Baruch Blumberg discovered hepatitis B virus when he wasn't even looking for a virus. And I say that to introduce you to one of a scientist's greatest assets, serendipity. Where does the word come from? It's an old Persian folk tale written about a king of a kingdom called Serendip, now thought to have been Sri Lanka, actually. And the king had 3 sons who were lounging around the palace, enjoying the luxury of being princes. And the king was so annoyed with this that he took them and said, look, you're going to leave the palace and we're going to set you out on a journey and you have to find your own way. And I want you to suffer as some of my other people do. I want you to learn what it's like to live outside the gilded cage of a palace. And off they went. And in the story, which was then translated in 1557 into Italian and subsequently to French and subsequently into English, the 3 princes managed to find their way only by happy accidents, by noticing things that they weren't originally looking for. And somehow those notices gave them the path in which they were to tread to achieve their goal. And this story was read by an Englishman called Horace Walpole in the 18th century, who was a writer, a critic, a literary figure, and also who wrote many letters. And he was so convinced of his own importance that he recorded all of those letters. And the word serendipity, he was the first to coin it, having read The Three Princes of Serendip, as a word meaning making discoveries of which one is not looking for. So chance discoveries. And the word has stuck in the English language since that. And of course, it took 200 years for Hollywood to catch up, but here is the film that followed it, John Cusack and Kate Beckinsale in 2001. And if you could read the strap line on top of the colored picture there, destiny with a sense of humor. So you can see, well, I'll lead you to see the film and see what you think of it. But certainly, Lady Serendipity was very important for Bloomberg. And he received the Nobel Prize for Medicine in 1976 for his almost by chance discovery. And if you're interested, he's written a book about that, The Hunt for a Killer Virus, Hepatitis B, which is well worth a read to understand how science sometimes works. The idea that a hepatitis virus, a virus that caused infection and cirrhosis in the liver, might also be linked to liver cancer was not seriously considered for several years after the discovery. But if you look, there's a clue here, that if you look in the top section of the slide, the pink countries reflect those countries with high incidence of liver cancer. And down below, the deep orange, those countries which have got high incidences of hepatitis B virus infection. Of course, this could be coincidental, or is it causative? That's always a huge issue to resolve when it comes to links between common viruses and malignancy. Let me introduce a second actor in this drama, which is Palmer Beasley, an American public health physician and vaccinologist who was spending the most part of his active career in Taiwan and looking at the effects of vaccination in that country on common diseases. And he was interested in the possibility that there may be a link between hepatitis B virus and liver cancer, but he actually conducted a survey which really showed the reality of the situation. He was following a cohort of Taiwanese government employees who were being followed over time and were looking at the incidence of hepatitis B virus infection in those individuals, and then following that and tracking them for liver cancer development. And he found that the rate of liver cancer development in hepatitis B virus positive people was over 1,000 per 100,000. If you look at hepatitis B virus negative people, it was way down by 200-fold to only 5. So clearly, there's very strong evidence that hepatitis B virus was a causative agent in many cases of liver cancer. So we can add to this to a table now, which gives us hepatitis B virus, liver cancer, and this is the global cancer cases per year in the year 2010. And you see the enormous global burden of liver cancer due to hepatitis B virus, 340,000. There is a second chronic infection of the liver, hepatitis C virus, which is likewise linked now to liver cancer. And I'll come back to this, these data later when we talk about prevention. So yes, viruses can act indirectly in specific circumstances. And hepatitis B and C viruses are the classic example of that. But what about another possibility, that the virus acts directly to promote the growth of infected cells and leaving its footprint in the cancer cells? Could that possibly be the case? And what would be the circumstances in that could happen? Well, certain viruses first need to make cells grow before the virus can replicate within them. Imagine a virus entering a factory which is on lay-by, which is not working. And the virus needs to replicate itself. It needs some of the cellular machinery to do that. So it has to wake the factory up. And it does so by driving growth transiently at the beginning of an infection. So here's a virus coming in, and it starts, the infected cell reacts by starting to grow. So that cell has become free. And then those cells are then capable of replicating the virus. And you see two of those cells now making virus particles. Where could this happen? What type of virus might do this? And we have an example, a very simple example. Everyone is aware of common warts on the hands or the feet, which are caused by human papillomaviruses 1 or 2. And this is a section through the skin, the normal epithelium, and you start at the bottom and cells begin in the basal layer and then move up towards the top to the outside world, and the mature cells slough off. So we're continually regenerating our skin. And this is a mobile system which can be exploited by papillomaviruses, simple wart viruses. They have a way of tracking in and infecting cells in that bottom basal layer. And because they need cells to be actively growing, they kick those cells into an extended life of proliferation. So the cells go on expanding. And as they get closer to the surface, they reach a stage where the virus takes over and begins to replicate itself, kill the cells, and you have what amounts to a wart. A similarly innocuous lesion, you might think, because we've all had warts and we all get over them eventually when the immune response controls the infection. But let's think about this in terms of what happens if one of those cells, if there's an accident within one of those proliferating cells and the virus is no longer able to replicate. Look at the dark green cell on the right-hand side there, which has not gone into replication. It's been, for some reason, unable to make the virus, and it's locked into permanent growth because something has happened there, an accident has happened. The virus DNA has been somehow altered, and the cell goes on proliferating, and as it proliferates, it can pick up bits of red genetic damage to its own DNA. And so the two are cumulative, and it could go on and finish up with a malignant growth in which the virus was the initiator. And sure enough, the question we're now asking then is, could this apparently innocuous wart infection somehow, in some circumstances, alter and produce malignant growth through the virus infection? Well, if that were the case, there's one important facet to take from this diagram, that the footprint of the virus is still in the cancer cells. And it was this principle that Harold Zurhausen took with him when he began to study the cause of cervical cancer in women. And he firmly believed that a papillomavirus, not the ones that cause viruses, warts on the hands and feet, but one of a related number of viruses could possibly be linked to cervical cancer in this way. He was doing this in the early 1970s when the idea was deeply unfashionable. If there was a virus, it certainly wasn't a simple papillomavirus. But Harold began by taking fragments of the DNA of known papillomaviruses and screening against the DNA of the cervical cancer cells, thinking that if there was a type of papillomavirus in there, he might pick it up, looking for similar DNA sequences to known papillomaviruses. And sure enough, that search was successful. And in 1983, his team reported that DNA fragments of a new papillomavirus type, which he then called HPV-16, because 15 types were already known, it was found in many cervical cancers. And this was a groundbreaking discovery, which proved not only to be correct, but deeply correct in almost all cervical cancers around the world. Either one, HPV-16 or HPV-18, another type was linked, and you will have heard of this through the popular press. And the viruses that cause that are indeed normal papillomaviruses that generally infect genital areas. That discovery by Harold was awarded the Nobel Prize for Medicine deservedly in 2008. And it's had huge consequences for public health when we talk about vaccination later. So yes, viruses can act directly or indirectly to cause cancer. And we can expand the table now and add papillomaviruses 16 and 18, and they're linked to cervical cancer, to the global burden. And look at the numbers, almost 500,000 in 2010 of new cases of cervical cancer per year globally. And of course, there are others. And briefly, I'm going to just say to you that there are a virus called human T lymphotropic virus 1, linked to adult T-cell leukemia, Merkel cell virus, linked to a rare skin cancer called Merkel cell cancer, and Kaposi sarcoma herpes virus, linked to a tumor called Kaposi sarcoma with about 66,000 cases per year. So you see the global burden is already increasing. What's very interesting about those 4 viruses is that they don't all belong to one virus family. They're completely different in their genome, in their biology. They only share one characteristic in common, and that is that they've got this capacity to transiently drive cell growth when they first get started on their infection. And that ability somehow gets abused, and it's not part of the normal outcome of an infection, but in rare circumstances, that growth promotion can be abused and come and permit the virus to enter a path which drives the cell towards malignancy. So there's one more, and you will hear from the introduction that it's a virus that I had been working on, and I'm going to tell you that story in the second half of the talk. But first, I have done enough lecturing in my life to know that there comes a point in a lecture, usually after about 30 minutes, when students are fidgeting, looking at their mobile phones, looking out of the window and thinking, oh my God, when is he ever going to stop? So the solution for that, I realized, I took from a friend of mine actually, is to show them a picture of mountains. So take a rest if you're okay so far. Take a rest with a view of Mount Everest. Let your mind just wander onto the blue sky and this magnificent landscape. You may like to reflect on William Blake's point. William Blake, a English poet of the Romantic era. And I saw this painted on a wall in a hotel recently, actually. I didn't know the quote. Great things are done when men and mountains meet. It was a quote in the Lake District, which is not really a mountain by any standards of the Himalayas. They're rather hills. But it reminded me of how I felt as a young PhD student and looking out at this wonderful world of nature, this magnificent biological system that we have, and wondering what part of the map am I going to explore? And not really knowing, and wondering if intuition and good luck and determination might lead me somewhere. And just to pursue the analogy with the view here, you'll see that the valleys between the peaks are covered in cloud. And often as a young scientist, and I hope there are some listening, you will have to descend into that cloud to make progress towards your goal. And very often in life, in the scientific life, you seem to be wandering in a mist and just wondering when you're going to come out, and whether your instinct and whether good luck will bring you out near a mountain that you would like to ascend, near a challenge that you feel is possible for you to attempt. And you have to first have an interest in that challenge. You have an interest in the subject. You've also got to feel that it is achievable, largely because of determination and technology which coming in, which could help you to address the essential questions. Is it sufficiently important for me to spend a lot of my life pursuing? Well, you get closer to the goal, and you're looking now at something that you may want to ascend. So look again at this picture. It's actually Mount Everest, but it could be any mountain that you might meet early in your career and thinking, well, that looks an interesting area to explore. I don't know whether there are any poets among you, but I'll read the quote from T.S. Eliot, which I've carried all my life. He is probably the leading poet in the English-speaking world of the 20th century. And he said in Little Gidding, "We shall not cease from exploration, and the end of all our exploring will be to arrive where we started and know the place for the first time." Something which intuitively may not make a lot of sense until you've experienced the situation where remembering how you felt when you set off on a journey, remembering how you felt, and then finally getting to the end, and you look back and you understand the part of the map that you've been exploring, and you understand why you started, and you understand in that moment the whole journey. Well, that's my interpretation of what T.S. Eliot meant. You may have others. Well, I hope you've enjoyed the Himalayas. I'm going to bring you back down to earth now, if anyone is interested, and talk about one more in this list. And it's the virus, which I said in the Professor Wei's kind introduction, I've been working on for, actually, I looked at the calendar the other day, almost 50 years. It was the first human cancer virus to be discovered. Before any of these were discovered above, it was discovered in the following way. Like a lot of stories, it starts in Africa. And this is a picture of Dennis Burkitt, who was born in Northern Ireland, lost an eye in an accident, lost his right eye in an accident when he was an 11-year-old child. Amazing. And yet kept his powers of observation and deep inquiry and deep Christian faith, actually. So in the Second World War, he served in the Royal Army Corps as a doctor in Africa. And when he came out of the Second World War, he decided to commit his life to work as a surgeon in Equatorial Africa, in Uganda. And he went first to Kampala, and then subsequently was sent way out into the bush, into the West Nile region of northern Uganda. And it was there that he noticed something which had escaped the attention of medicine in other parts of the world. It was a tumor which presented as a tumor of the jaw, usually in young boys, sometimes in young girls of the abdomen, of the ovaries. And he realized that these were all same manifestations of the same childhood cancer, which was extremely common in parts of the country. It was a cancer of lymphocytes, of blood cells, white blood cells. And this is a picture of the cancer under the microscope. And the round cells are the lymphocytes. And they're surrounding these little white boxes, which are basically debris. So this is a cancer with a very particular appearance under the microscope, which distinguishes it from all other types of lymphoma, of cancers of lymphocytes. Well, leaving the blue skies of Africa behind, we come to the grey mists of London in 1961, at Middlesex Hospital, where Dennis Burkitt came and gave on the 22nd of March a seminar to a combined medical and surgical staff meeting about the commonest cancer of children in tropical Africa, a hitherto unrecognized syndrome. It was at 5:15, not probably a very popular lecture, but it was attended by someone who became my colleague over 40 years and who was so taken with the story and realized its significance that as he walked out of the lecture theater, he took the notice down and kept it. That person was Tony Epstein, who was at that time, a relatively young pathologist. This is a later picture when I was working with him. A young pathologist who was trained in electron microscopy, the new technique of virus hunting, and decided that he would pursue the possibility of a link between viruses and human cancer using the electron microscope as a tool. He was a deeply unpopular kind of activity in those times. Most people thought he was chasing rainbows, but he listened to Dennis Burkitt and thought, my goodness, this is a cancer which, and these are the maps which Dennis Burkitt had by that time produced of case reports of this cancer within equatorial Africa. And you can see that the outcome of that safari looking for distribution of the tumor covered an area which was almost exactly the same as the area covered by holoendemic malaria, a parasitic infection caused by mosquito bites carried by the mosquito. And it immediately raised the possibility that this cancer was caused by a virus that could be spread by mosquitoes. Could that be correct? So as a result of the lecture, tumor samples were flown on ice from Kampala in Uganda to London Heathrow and to Middlesex Hospital, where Tony Epstein started to look at those samples under the electron microscope. The first 25 samples came in 1962 and throughout 1963, and he looked at the cancer cells and saw absolutely no viruses. Oh my goodness, this is not good news. And quite often you reach the end of your tether as a scientist and say, okay, this is it. I can't really pursue this project. It's probably the end of this project, and it's probably the end of my career as a serious scientist. But then on the 5th of December, 1963, the 26th sample arrived, not direct from Kampala. It had to go to Manchester because there was fog at London Heathrow Airport. Eventually it arrived by truck coming down from Manchester, and it looked rather different from anything they'd had before. Instead of just little fragments that grow in saline, it was very cloudy. And the first assumption was, like any sample you get in the lab, if it's very cloudy, it's bacterial infection, and oh, let's throw the thing away. We're not going to get anything with this. But Tony and his team put the cells under the microscope and saw that this was not bacteria. These were living cells from the tumor. And he put them in artificial medium in a 37-degree incubator, and they started to grow. And this was the first time, as it happens, that any human cancer had been grown in cell culture in the laboratory. Of course, many now are in one way or another. But this was the first example, late 1963. And within a few weeks, they had enough cells growing that they could commit some to a pellet and prepare it to look in the electron microscope. And they did that. And the first time they looked, they saw in a very small fraction of these cancer cells, virus-like particles, not just virus, but particles that looked exactly like a family of viruses that were just beginning to be known, herpes viruses. And you'll think about herpes simplex virus and cold sores as being the first such example. This was obviously a herpes virus. growing in some of the cells of the cultured tumor. Well, the work was published in The Lancet in 1964 by Tony Epstein, his colleague Bert Achon, and Yvonne Barr, his research assistant, who had actually done the cell culture. And this picture was published, and to general disbelief, saying, oh, well, you found a virus in cancer cells, but actually it looks like a herpes virus. And we know that herpes viruses grow in many cell types, and it's almost certainly a contaminant that's got in your cultures. So this is the criticism and general skepticism that this story had to deal with after the first publication. But not everyone was so skeptical. And this is Werner and Birgitta Henley, a team of virologists working in Philadelphia who had been working on influenza and other viruses and developing antibody tests that can detect new viruses. And they set up an assay, which was called a fluorescence assay, that under the microscope, after you combine a serum from an infected patient with a cell preparation in which some of the cells are making the virus. You can see the green dots here are individual cells that are making the virus in this culture. So this was the first test of antibodies against this so-called new virus. And by having this test, the Henleys could now go through a whole batch of sera and say, well, having this antibody, does it correlate with having antibodies against any other known herpes virus? And the answer was no. They had serum which recognized this virus, but did not see any of the other known human herpes viruses. So yes, this really is a new virus. Question, how widespread is it in the human population? Is it restricted to equatorial Africa? Is it very rare in most populations? Answer, no. It's a common virus in all human populations. All human populations. The majority of people are already infected with this, which really fed the skepticism, saying, well, really? Are you telling me that a virus which is widespread in every community is linked to this particular cancer? Certainly, it hit the idea on the head that this was a cancer caused by a virus spread by mosquitoes. But what about the other possibility? That it was holoendemic malaria was the cofactor. In other words, being bitten by a malarial carrying mosquito 12 months of the year, which was what happened in these equatorial regions. Could that be a cofactor? Well, there was an important clue that there was one other area of the world which had equatorial-like conditions, and holoendemic malaria was present. And that was in the lowland areas of New Guinea. The coastal areas of New Guinea had the same conditions, and the same tumor was found there, and the virus was found associated with that tumor too. So holoendemic malaria is a chronic immune stimulus driving growth of the cell type from which Burkitt arises. That's the first factor. But what about EBV? Was it just a chance that Tony Epstein had found it in an African Burkitt lymphoma? No. When you develop tools to look at footprints of the virus, and remember the principle, if a virus is involved, it will be there in the malignant cells. EBV DNA was found in those cells, and the green picture here is one viral protein which is being made in every cancer cell. So every African and every New Guinea Burkitt carries these footprints of the virus. So it's too much of a coincidence to ascribe to an implausible situation. The virus is present and it's active in every tumor cell of this type. And we now know how it's working, that it's driving cell growth to some extent and increasing cell survival, the most important factor in this whole story. But a virus infection is never sufficient on its own. There have to be additional genetic changes. And I just want to mention one because of its historical significance, that when people looked at the chromosomes of this tumor. Now, we have 23 pairs of chromosomes in our normal cells, and here are the chromosomes from a Burkitt lymphoma cell with a very characteristic change. If you look at the bottom left-hand arrow, you will see that the two pairs of chromosome 14 members there, one of them is shorter than the other. And that's because a little bit of that chromosome has been lifted off and translocated by accident onto, look at the top yellow arrow, one of the pairs of chromosomes of chromosome 8. So there's been a genetic accident, a chromosomal accident, which is extremely rare. But when you look at it at the level of DNA, you can see that that accident has led to a key cell growth promoting gene being switched on permanently. So we know that a rare chromosomal accident in one single cell has been the clincher that drives that cell into uncontrolled growth. But that accident has happened in a cell in which EBV is already driving it. So 3 factors, 1 cancer. And this is probably the best example of a multifactorial cancer development program that was first identified. So Burkitt lymphoma has been a very important Rosetta Stone, if you like, for understanding multi-stage carcinogenesis. So there we have it. This first EBV-associated cancer is endemic Burkitt lymphoma. And I just want to, although there are many to come, I'm just going to talk about one, which is of particular interest and much closer to home for you Hong Kongers. So that story, again, begins in Africa. And it begins with another clinician and how important clinical observation has been in this whole story. Peter Clifford was an ear, nose, and throat surgeon working in the Kenyatta National Hospital in Kenya in the 1960s. And he helped the Henleys to try to answer a question of whether antibodies to EBV, to the virus linked to Burkitt lymphoma, were different in Burkitt patients to controls. So he took serum from some of his Burkitt patients coming in, and he took serum from other patients that came to his clinic. He was an ear, nose, and throat surgeon, so his controls were patients coming to him for surgery with cancers of the head or neck. And a third element was Lady Serendipity, someone to whom you've already been introduced. And why is she important? It's because African patients do have unusually high antibodies. This is the result. And the controls, patients with other head and neck cancers, do not. But except for patients with one other tumor that was part of his clinic called nasopharyngeal carcinoma, a tumor that he saw in East Africa reasonably commonly, but was very rare in the West again, but was very common, known to be common throughout Southeast Asia. And here is the worldwide distribution of age-standardized incidence rates, and you see the concentration of dark areas around South China and around the Malay archipelago in the South China Sea. So we'll come back to this map later, but essentially, we know that this was a worldwide tumor, particularly known in southern China and in Hong Kong. And this is what's really rather sinister about this story, that the tumor arises in the lining of the nose, in the epithelial cells that are lining the postnasal space right at the top of the nose. But it quite frequently presents much later. So it may cause a bit of irritation in the nose, but you never know about it, or it's put down as some kind of rhinitis. And then the lumps appear in the neck, in the cervical lymph node, because the tumor cells have spread down in the lymph, down into the lymph node. And quite often they present with this serious metastasis, at which stage, certainly in the early days, it was very difficult to treat. So that's the background. Now, why do we have this concentration among people who are, in inverted commas, Cantonese, Southern Chinese people? Is there some genetic susceptibility? And we know that family members of NPC patients are at increased risk versus their neighbors in the same community who are unrelated to their family. So this is already an important demonstration that nasopharyngeal carcinoma is, to some extent, a genetically susceptible disease. And we now know many of the markers which are distinguishing high risk among the Chinese population. So it is, yes, it seems genetically susceptible, but what about the other Southeast Asian hotspots? Could they be related in some way? So this is where we begin to look at the map again, and I introduce you to a friend of mine called Joseph Wee from Singapore, and the initiator of the so-called Wee hypothesis. This is what I call it, and it relates to a story that a proposal he made in 2010, and call it, is NPC really a Cantonese cancer? Now, Joseph is a great aficionado of ladies' fashion magazines, quite obviously, and he sent me these delectable pictures of young women in traditional dress, and people from Borneo, from one of the ethnic groups of mainland China, and the ethnic group of Taiwanese, and also very interesting, even in North India, people of Southeast Asian origin form in Nagaland a population which are again at increased risk of NPC. And Joseph's idea was the following, that if you look at the map on the right, the green areas, you see the contours of land as laid out in the South China Sea as we know it today. But go back 20,000 years, and here on the green areas on the left map, you see the land mass as it was before the end of the Ice Age, before sea levels rose. And go back to the right-hand map now, the sea came in and submerged the land in the light blue and formed the contours of the land, which we now know today. So Joseph's idea is that in this original land on the left, there were people called the original Bai Wei people with a high risk of NPC. And with the isolation of land masses, those people have separated. But that diaspora of the Bai Wei population all carry high NPC risk. And that is why we get this kind of distribution of high incidence within Southeast Asia. There are other areas of high incidence that have to be dealt with separately. And Joseph has some really crazy ideas as to how that could have happened, but I'll pass over that for the moment. So there is genetic susceptibility of an Aboriginal Cantonese people, let us call them. Yes, but is it the whole story? Here's another player in the field, Philip Well, another non-scientist, some was an epidemiologist, working on public health in the State Department of Health in California. Why was that an important situation for him? Because he was interested in the relationship between genetics, lifestyle, and disease susceptibility. And in the 19th century, of course, California was a mecca for large amounts of Chinese migration to areas during the gold rush, and the creation of ethnically Chinese populations, which persist now to this day within California. And he wanted to know how that migration had affected their cancer risk, because he realized that migrant populations such as the Chinese create ethnic groups which keep their genetics, but gradually change their lifestyle from the Chinese diet, let's say, to, I'm not going to say McDonald's, but I'm going to say something different with the move to California. Well, he noticed something very important, and this is one of the first of several papers, this one dating back to 1965. he found that first-generation Cantonese migrants to California had the same NPC risk as the brothers and cousins that they had left behind in China. Now, these young men principally had spent their childhoods back in their native land before migration, and they carried the same NPC risk. But their second generation, their children, and their grandchildren in the third generation gradually reduced their NPC risk incidence up to tenfold even higher in some studies. But even in the third generation, their risk was still tenfold higher than their Caucasian neighbors in the state of California. So it shows that genetics is not the whole story. There is some lifestyle or environmental factor linked to being Cantonese. What could it be? Here I introduce you to someone who may be known to many of you, at least historically, and that is a giant of medicine in Hong Kong, Professor John Ho, sometimes called Emperor Ho, who I was honored to meet in 2002, just 3 years before his death, while I was working in Hong Kong. And he had immense influence on the development of cancer services in Hong Kong. He set up radiotherapy, a key component in treatment for nasopharyngeal carcinoma. He was an academic leader and administrator, but also intensely curious, particularly about a question as to why NPC was so high in Hong Kongers. And he looked to the boat people. Here's a picture of them as I used to live in Aberdeen Harbour in Hong Kong. And their incidence is double that of Hong Kong Cantonese. They are in fact, and Joseph Wee says this, that they're probably the closest one could get to the original Bai Wei people with their high NPC incidence. And John Ho looked at their lifestyle, and particularly their lifestyle for young children, and realized that they had a potentially very dangerous habit of weaning their children onto a particular salted fish concoction. And looking hard at this salted fish in the way it was made by the boat people, there were carcinogens in that. There were dangerous chemicals which either could be swallowed or inhaled by the young infant over many years and could initiate cancer change very early in life, just the starting point of the process. And the more people have looked at the epidemiology of salted fish consumption, they do find that it is linked to NPC risk, certainly among Southern Chinese people. So John Ho's observation is validated, and that is one of many reasons why he is remembered as the father of NPC research in Hong Kong. What about Epstein-Barr virus? Is it in the tumor cells? Remember the old mantra, if the virus is doing anything, then it should leave its footprint in the cancer cells. It should still be there. Now, that was a particularly important question because up to that point in the 1970s, the virus had only been seen to infect one type of cell, a particular class of lymphocytes. This is a slide of the appearance of a nasopharyngeal cancer tumor. And you see that the black dots are the invading lymphocytes, but the really malignant cells are the flat, purple, lighter cells, which are dominating the center of the picture. These flat epithelial cells are the cancer cells, but they're not the cell type that normally EBV would infect. EBV likes lymphocytes. So could the virus be there? Well, you can look for the DNA, and now you can look for the proteins. So this is a slide made by a friend of mine staining for one of the key EBV proteins, which is expressed in those cancer cells. And indeed, every case of nasopharyngeal carcinoma worldwide carries the viral genome in every cancer cell, and it's active and making cell proteins. So the virus is an essential factor. And it doesn't matter whether you're a rare case of this particular kind of NPC in the US or Europe or in Africa or here in Southeast Asia, the same virus is always there. So we can add a second cancer, a very important one in terms of its global case numbers, 10 times more common than endemic Burkitt lymphoma, for example. And so we now know the virus is really dangerous and could cause cancer in at least 2 different cell types. And those cell types expand actually. So I don't have time to tell you about more of this, but to say that there are now 7 tumors which are linked to this virus. And you get the percentage of tumors which carry the virus. So Hodgkin lymphoma is probably a disease you've heard of. About half of all cases of Hodgkin lymphoma carry EBV in the cancer cells. The other half have arisen by a different route. Gastric carcinoma at the bottom, that's stomach cancer. Now, between 5 and 10% of all cases of stomach cancer worldwide carry EBV in every cancer cell. And that's a distinct subset of stomach cancers, which is genetically distinct from all the rest, and also distinct in the sense that EBV is there too. So you get some idea now of the protein, the many sides of EBV and its associated diseases. There are others that I could speak about, but I want just to focus right now your attention on the global impact of this virus in terms of the number of new cancers per year that it causes. And let's put that now briefly at the end in this larger picture of all virus-associated cancers, with EBV being at the bottom, being associated with 7 different types of cancer. Now, what are we going to do about this? That's the major question. And we were already set on the path by Barack Bloomberg with his vaccine, the vaccine that was developed against hepatitis B virus, where developing this vaccine and administering it is bringing down, is essentially blocking hepatitis B virus infection, bringing down cancer rates. Hepatitis C virus, there is no vaccine, but there is now a battery of drugs which can control hepatitis C virus infection, and that will bring down, because it blocks the chronic nature of that infection and the chronic damage to the liver, that will also bring down radically the number of cases of liver cancer that arise because of that infection. We already know from Harold Zurhausen, his work was followed up by the development of an HPV 16 and 18 vaccine, now being given in many countries, I'm sure, including Hong Kong, to young girls, and prepubescent girls, and that will essentially already bringing down, even within 10 years of the initiation of the vaccine program, it's bringing down cervical cancer rates. It has the potential to essentially eliminate cervical cancer, a hugely important global health goal. Nothing yet against the other 3 viruses that I told you about, nothing yet against EBV, but there's a lot of work going on to develop an EBV vaccine which could prevent perfection, or could control the infection in a way, or could even target the cancer cells if they ever developed. Or one could go through the other route, and there's a lot of work going on now, looking at drugs that might interrupt the action of the virus proteins that are expressed in particular cancers. So there's much work to do. Let me just end by stressing that Hong Kong is one of the leading centers for NPC research worldwide. And that's why I've been a missionary coming to Hong Kong, a disciple following these people for many years and working with them as much as I can. George Chow and Maria Lung and KW Lo from Hong Kong U and from Chinese U have done much to understand the genetic changes occurring in NPC, and also much to show how those changes interact with the virus to cause the cancer. On the bottom line, we see Anne Lee, Anthony Chan, Bridget Ma, all clinical oncologists who are leading the fight in new treatments and new trials to improve the fate of nasopharyngeal carcinoma patients. And big strides are being made there, of course. And finally, I want to give pride of place to Gary Wong, Professor Gary Wong, of your own Department of Chemistry, who is a drug designer and who is doing very interesting work on the drugs that could intercept the action of EBV proteins. So you're sitting in a place which is actually a leading center for research on this virus and on this cancer. So we've come to the end of the story, and I hope some of you will begin to know the place for the first time. Before I go, just one terribly, unforgivably self-publicizing slide. If you liked that story, you might try this. Cancer Virus is a book about the EBV story, which I wrote with Dorothy Crawford in 2014. It has not yet been made into a film. If anybody you know, any Hollywood publishers, please give them my email address. In the meantime, as a student said to me the other day, it's also available at greatly reduced price on eBay. Do not buy it from OUP. Well, that's it. That was the story that I wanted to give you. Thank you for listening. Thank you for your attention.
Thank you, Professor Rickinson, for the very insightful and lecture, and also some very encouraging and motivating words for our students. So it is now open for question and answer. So we would invite a question from the floor, and then we will continue, followed by a question from online audiences. Any questions? Okay, there's one, please. You can turn on your microphone.
Hello, Professor Rickinson. My name is Serena. I'm the RPG student of Dr. Long. When I first got to Dr. Long's lab, I was naive to think that we can target a particular cellular pathway to stop all type of cancer, and I know it was very naive. But then I was inspired by the idea of how we humans manipulate our genetic contents to stop disease from transport from parents to their children, and then also inspired by how we manipulate our genetic contents by using mRNA vaccines to treat the COVID-19. And so recently in these few months, I was thinking about how about we manipulate our own genetic contents again, and to force cancer cells to eject their nucleus, like how human red blood cells eject their nucleus, to decrease the lifespan of the human cancer cell, and then eventually end the cancer. I'm thinking about this for a few months, and I'm wondering if in the near future that it will be possible.
So you're asking whether you could manipulate a cell to eject its own nucleus?
Yes, just like how human matured red blood cells does. Because I think that if we know more about how this process actually begins and how it's processed, then we can manipulate that pathway and apply it to our cancer cell, and then we trick the cancer cells to force out their nucleus, and then it will be much easier to treat the cancer.
Yes, well, that is a major question. And I must say, I haven't thought it through. The whole process of nuclear ejection starts to be very complicated. Of course, it happens in erythrocytes, is what you were saying, I think, in red blood cells as they develop. But that process, I don't know how much that is understood. I think probably what you're trying to do more is more achievable is to try to induce death of cancer cells using a natural process. All right, rather than nuclear rejection, there is a process called programmed cell death or apoptosis, and it is now intensely studied, and there are genes which activate that process in particular cell types. And so this whole business of driving programmed cell death, driving that program, which is a normal biological program. When cells die, they actually actively die. They decide and they are triggered, and they will die by a process of dissolving the nucleus and cutting their DNA. So it is a process that goes naturally, but what one needs to do is harness it and activate it at the time when the tumor cell becomes self-governing, if you like. So that is the hope of a lot of cancer researchers, actually, to use the apoptotic machinery and drive cell death. So I think that's more achievable, and that's probably an area that you could think a little bit more about.
Thank you. Thank you very much.
Okay, thank you. Any other question from students?
Hello, Professor Rickinson. Thank you. Actually, I was doing something we have a drug, and this drug is to induce the lytic reactivation. So essentially, we aim to drive the virus out of the cells. And after that, we hope that the immune system could do something about the cancer, because kind of to alert the immune system and then to attack the cancer. So I refer to one of your early studies in which you identified that the cytotoxic T cells, there's a memory cytotoxic T cells in the blood from the EBV positive donors. And those cytotoxic T cells, after re-challenge with the EB virus, then the memory T cells can eradicate the B cells. So in that study in the 1970-something, have you also studied the helper T cells? Because you only focus on the cytotoxic T cells, but if we're interested in the helper T cells to see if any of the cytokines will be released, and after which could it be used to stimulate the other effector cells to eventually kill the cancer cells?
Yes, I understand the question. I haven't talked about my work in immunology, I have to say, but obviously you've seen something of it. So yes, we have, the main attack dogs of the immune system, you might say, are so-called cytotoxic T cells, CD8 T cells. And we concentrate a lot of our early work on that. But there is another panoply of attacks which are made by helper cells. Sometimes those helper cells are secreting cytokines, which can activate killer cells. Sometimes they be killer cells themselves. So the more we look at it, actually, these helper cells can also be cytotoxic after they're activated. So they have a double-edged attack mechanism. And so we want to try and, let's say in vaccination, if you want to improve the immune response against the virus, then you would need to activate both helper and cytotoxic T cells, both the CD4 and the CD8 cells. And there's a lot of work going on in just that process. And it's quite interesting that the range of targets that the CD8 cell, the traditional cytotoxic cell, recognizes in EBV are not the same as the targets that the CD4 cells tend to attack. So the relative order of antigenicity of targets, of useful targets, is different for the two types of attack. So by combining them, you could double up and get maximum effect. So of course, we're still very interested in that, because the whole question of developing a vaccine that could prevent infection with virus requires a study of how T cells work, and also you could use the same T cells of particular specificity to attack the cancers like nasopharyngeal carcinoma. So both areas of work are going on. How can we use T cells in a traditional, what's called prophylactic vaccine? In other words, a vaccine that could give you immunity against ever becoming infected with the virus. You could kill the virus straight off when it first comes in. So there's that kind of vaccination. And there's therapeutic vaccination where you're trying to drive the immune system, the patient's own immune system, to recognize bits of the virus which are present in the cancer. All of these sound easy when I say it like that. And of course, they're intensely difficult because there are all kinds of obstacles to that. And cancers learn how to evade such responses and so on. So yeah, your question is very timely, and it's something that a lot of people around the world now are really studying intensely.
Actually, in your earlier study, the work that you identified that the memory T cells, this study was done in 1970 something. I read it previously because now we are trying to really study the helper T cell, see if it could be activated, because we already have the drug and we are doing some research to check the cytokine. But so far, the work is kind of like what you mentioned, like in the mist. We have this hypothesis, but the support, the evidence is not very much strong. So as you have done relevant kind of similar work with address on the cytotoxic T cells, I was wondering, actually in that work, have you ever tried to look at the cytokine production at that time? And are you optimistic that the memory T cells, the memory helper T cells in the body, in the circulation, is good enough to do the work, to do the job? In other words, can you give some suggestions on such a pursuing of the utilizing the memory helper T cells for cytokine production for the therapeutic of cancer?
Yes, well, we have characterized cytokines as much as we knew. We have looked at multiple cytokine types, but there is a panoply of cytokines, and there may be others that we still haven't recognized. But certainly, the range of cytokines that helper cells produce contain within them potential damages, things that could attack infected cells. So it's not just the cytotoxic killing mechanism that's important. There's also these chemical messages which are coming out. So if you email me, we can go into a detailed discussion of this.
Sure.
And come out of the mist and get the clearing, yes?
Okay, then. I will send you some kind of the preliminary results. Thank you.
That's very good. Thank you.
Thank you. There are a few questions online, so because of in the interest of time, we pick a few that we find interesting. So there's one question from this student, PhD student from Department of Clinical Oncology in HKU. His name is Michael Chong. He asked, first question, since EVV can infect B cells, is it possible for the EVV exosome within the MPC TME, which I do not know what is that, to affect B cells? Okay.
Is Michael there? I can't quite.
I think he asked, I would rephrase for him. I think he asked if he assumed there is exosome, and he asked if the EVV exosome would play a role in affecting the B cells. Okay, tumor microenvironment. Okay, he just explained.
Yes. So this whole issue of exosomes carrying the virus or carrying viral proteins is a very active part of current research. And it's still not clear to what extent this is important. It is actually, while we're thinking about it, one of the mechanisms whereby the virus could spread from its natural infection type, let's say a B cell, and get into other cell types that are in the vicinity, which would not normally be infected. It could smuggle its way inside an exosome. That's one possibility that we have thought about without any serious serious advance. But if you think about, how would the virus get into a stomach cancer, so stomach cell, into the epithelial cells lining the stomach? It should not be there. And so it may be that when you get inflammation of the stomach, for example, for another reason, perhaps with Helicobacter or something else, you may create an environment in which you have an invasion of EBV-infected B cells, yes, and they could spread through exosome release or through some other mechanism, the virus into a cell which is not a natural target. We still don't understand how the entry is working. And exosomes are one interesting possibility in that context.
Yeah, I agree, because the exosome is really tiny. I think it's possible that they just pick it up through some endocytosis.
Yeah. You see so many questions that have not yet been answered.
Yeah, that's right. So we have one last question, if you would allow me to allow that question. To what extent do you think, sorry, this is from, what is his name? Okay, sorry, I cannot, I think it's Emma. To what extent do you think scientists today have the freedom to explore and experience serendipity, okay, given the funding limitation, grants competitiveness, and often risk senses of funding bodies today? Okay, I think what he or she meant is very challenging, competitive era that we are having now.
Yes, Emma, this is a question which has been asked of me many times in my career. And it's a problem we all have to deal with. And it is an issue that I and others in the field are desperately trying to correct, to make funding available, not just for big groups, but for people with interesting ideas. It's difficult to get a foot on the ladder. And what I could say is perhaps try to build a case before you go for application. Try to build a case from some preliminary evidence to give people the confidence that this is the kind of hill or mountain that is worth ascending. That is a real possibility. And so that would be my general advice, to try to build the case as strongly as possible. But don't be deterred, because I remember going back 30 or 40 years sitting on grants committees and people submitting grants to saying, oh, well, we don't need to use normal T cells. We can genetically engineer them to be killers with a different recognition mechanism. And why don't we make a chimeric antigen receptor T cell? Something which looked like science fiction 40 years ago, but was conceivable, and now is a reality in the clinic. So don't dismay. Keep thinking of possible blue sky approaches, then try to build up as much of a preliminary case as you can and go for funding at the right time.
Thank you for the encouragement. So I have this question from a very eager PhD student who asked, may I ask Prof. Rickinson giving some general suggestion for the PhD students? So this would be the last question from the student. I think a lot of students asking very similar question. So suggestion maybe how to be successful or complete their PhD.
I'm not sure I'm the best possible. Let me tell you this story. I took a PhD studentship, a project for which I was distinctly unqualified. It was trying to develop a type of machine by exploiting electron microscope to irradiate minute particles within cells, minute organelles called the nucleolus. I can't imagine a crazier project, or I can't imagine a student who was less qualified to do that project than me. And I started, and after 18 months, I nearly gave up. I honestly was in the point of saying, this is not for me. So students who are 18 months in without a result, don't despair. There is a way out. I was lucky to discover that you could do the same job using a drug. You didn't need to do this fancy microirradiation. And I switched to the drug and I got a PhD in the end, only just. So I want to stress to you that no matter how bad things are, don't give up and try to take advice, try to ask people. Generally, if you're on a project which looks difficult, don't be afraid to talk to people in the lab next door. Don't be afraid to email somebody in another country and saying, I'm a student, help me. Because everyone who is senior in the field, we've all been through it. We've all been through this crisis of wandering in the mist, not knowing. And there is help there. And generally, people who are senior now, have been through that, are very willing to advise and help. If you're serious about being a scientist, don't give up.
Yeah, totally agree. I just said the same thing to my student today. I said not giving up because he was in doubt with himself. So I think it's something that all of us will go through. Right? And you will make it through as long as become very determined of what you want to do. Okay, so thank you. So before the end of the lecture, we have a group photo taking session. So may I invite Professor Alex Wai, Professor Rick Wong, Professor Yike Guo, Professor Jiming Liu, and Professor Calvin Leung, and colleagues to take a group photo with Professor Rickinson. Okay, thank you, Professor Rickinson, for your very inspiring sharing again and encouraging our students. And I would like to also take this opportunity to thank all of you for your being here today and very active participation. And wish you a very nice evening in Hong Kong and very good day in UK.